Hi there, in this video we are going to discuss TP53, also known as the guardian of genome. So TP53 is also a tumor suppressor gene. It means that it suppresses the cells which are faulted in any way from growing and dividing. In this way it suppresses the tumor formation. So TP53 is a tumor suppressor gene and P53 is the protein product of the TP53. So this protein product p53 is basically a transcription factor and it is short-lived. Its half-life is about 20 minutes, and what happens, it is basically bounded to MDM2 protein, and this protein leads to the destruction of the p53. So what happens when there is some mutation in cell, or cell is stressed in some way, there is post-translational modification in p53 protein, and this post-translational modification is driven by the product of another gene, which is ATM gene, and this post-translational modification frees p53 from MDM2 and increases its half-life. So what we want to say, p53 is a protein product of TP53. It is a transcription factor. Its half-life is usually 20 minutes, and it is bounded to MDM2 protein, and this protein catalyzes its leads to its destruction. However, when cell is suppressed in some way, or there is some mutation or some DNA damage, what happens, ATM2 gene product becomes active and it leads to the post-translational modification of p53 protein, and when it becomes modified, it becomes free from MDM2 and its half-life is increased. Okay, this TP53 is guardian of genome, and it can lead to three fates of cell. It can lead to permanent arrest of cell cycle, which is called quiescence, or it can lead to temporary arrest of cell cycle, which is quiescence, or permanent arrest of cell cycle, which is senescence, or it can lead to the programmed cellular death, which is apoptosis. It can lead to cell to follow one of these three fates, either quiescence or senescence or apoptosis. We previously studied retinoblastoma. Retinoblastoma was responding to external signal, which were either growth stimulatory signal or growth inhibitory signal in the form of epidermal growth factor, or platelet derived growth factor, or transforming growth factor, and so on. However, p53 responds to the internal stressed signal of the cell. Okay, and it senses any damage to DNA or internal stress condition, for example anoxia, etc., and stimulates the either arresting cellular growth or destruction of the cell. Okay, so p53 mediated cell cycle arrest occurs in a way that when there is cellular stress, there is activation of gene which is CDKN1A. This gene becomes active in the stressed condition, or you can say when there is DNA damage. There is some DNA damage. This DNA damage activates the gene CDKN1A, and its protein product is P21. Okay, so its protein product P21, what it does, this P21 basically inhibits cyclin D and CDK complexes, or you can say 4-6 complex, which we studied in the retinoblastoma or RB lecture. So this cyclin D or CDK4-6, what it was doing, it was basically leading the phosphorylation of the retinoblastoma or RB. So when retinoblastoma protein product was getting phosphorylated, it was becoming inactive, and when it was becoming inactive, it leads to the cell cycle or cellular division. However, what happens when cell is stressed or there is some DNA damage, CDKN1 gene become active. Its protein product was P21. This protein product inhibited cyclin D and CDK complexes, and when this complex was inhibited, there was no phosphorylation. Normally it leads to phosphorylation, but when it got inhibited by P21, there was no phosphorylation, and when there was no phosphorylation, retinoblastoma remained hypophosphorylated, and In hypophosphorylated state, retinoblastoma is active. Active state of retinoblastoma is hypophosphorylated state, and when it was active, it holds EGF transcription factor and cyclin E production is reduced, and there is the arrest of cell cycle. So in this way there is seizement, or you can say stopping the cell cycle. So DNA damage activated CDKN1A gene whose protein product was p21. This p21 inhibited cyclin D and CDK, and when it got inhibited, there was no phosphorylation, and when there was no phosphorylation, retinoblastoma remains hypophosphorylated, and hypophosphorylated state of retinoblastoma is active state, and in its active state retinoblastoma stops the cell cycle. So in this way there was a temporary block in cell cycle or quiescence. Now p53 protein upregulates DNA repair mechanisms. Okay, on one way it stopped the cell cycle, on the other way it upregulated DNA repair mechanisms or DNA repair proteins. When DNA repair protein became active, they tried to repair the damage. If damage was small and its repairing was possible, cell will again enter into the cell cycle. However, if the repair was too much, supposedly repair was done, what will happen next? Then p53 protein will upregulate MDM2 protein, and when this protein will be upregulated, what will happen? It will lead to the self-destruction of p53. Okay, so p53 on one side stopped the cell cycle by interacting with retinoblastoma. On the other hand, it upregulated the DNA repair mechanism. If DNA was repaired, then it upregulated MDM2 protein, and this protein led to the destruction of p53, and everything returned to normal and cells again entered into the cell cycle. However, if the damage was beyond repair, what will happen? Cell growth will be permanently arrested. Permanent arrest in cell growth, and this permanent arrest in cell growth is by global changes in the chromosome. Global changes, I can say, our chromosomes are changed or altered in some way, and CDK inhibitors are activated, and there is permanent stop in cell cycle or senescence. Next is apoptosis. If damage is too much and the cell is not suitable to allow the cell to live, then it activates pro-apoptotic genes, which include BAX and PUMA, and these pro-apoptotic genes lead to the apoptosis of the cell. So it is the normal functioning of TP53 gene or tumor suppressor gene. However, if its function is mutated, normally it is not allowing the cell with damaged DNA to progress and grow and divide. However, if it is damaged in one way or other, what will happen? There will be no arrest to cell cycle, there will be no DNA repair, and cell will continue to grow. And when cell is mutated DNA will continue to grow. What will it lead? It will lead to the production of tumor cells and carcinogenesis. Okay, so it is highly necessary to guard the genome of the cell. However, if it is not present, genome can be damaged, and there will be no repairing and no rest in cell cycle, and it will lead to the production of tumors. So one terminology we need to know here is Lee-Fraumeni syndrome. What is it? There is also two alleles of the TP53 chromosome, and for carcinogenesis both alleles should be absent, and which is also Knudson to hit hypothesis. However, in a Lee-Fraumeni syndrome, it is a familial condition in which one gene is mutated genetically, and in such it is called Lee-Fraumeni syndrome. In such case, only one mutation or mutation in other allele is required to lead to carcinogenesis, and such person are predisposed to a variety of carcinomas, and they develop cancer at an early stage, and so on. They can lead to different kind of cancer, including breast carcinoma, soft tissue sarcomas, and so on. And the oncogenic viruses, for example, human papilloma virus and hepatitis B virus, they can target TP53 and lead to its inactivation, can lead to carcinogenesis. Now we view the diagram of TP53 from book also. This is the diagram of TP53. You can say this is a normal cell, and there it is stress stimuli in the form of DNA damage or hypoxia. what happened, p53 got accumulated and it bound to DNA and it led to the transcription of multiple genes. And what happened, on one side it induced p21 product of CDK gene or CDK, and this is the protein product of CDK N1A protein, N1A gene. So it's a product. What it did, it lead to the arrest of cell cycle at G1 S phase by interacting with retinoblastoma. And on the other side, GADD45 gene got activated and DNA repair mechanism got activated, and there was a repairing in the cell cycle. And if cell got repaired, they became normal cell and enter into the cell cycle again. However, if damage was beyond the repair, they enter into permanent growth arrest, which is senescence. And if there's too much damage and it is not suitable to let the cell live, then VAX gene or apoptotic gene got activated and it led to the apoptosis or death of the cell. However, if there is mutation in the p53 gene, then DNA damage will not be sensed and there will be no repair response or senescence or apoptosis. And what it will do, it will lead to the mutant cell which will continue to grow and it will lead to the malignant tumor. So it was all about today's lecture. Thank you.